Hello everyone, welcome to ITLS Academy. Our today's lecture is using antimicrobial chemotherapy to control microorganisms. We will start our lecture with objectives. We will see what are the objectives of this test. So, after completing this exercise, the student will be able to perform the following objectives. Number 1. Antimicrobial chemotherapeutic agents. In this objective, define antibiotic, antimicrobial chemotherapeutic chemical, narrow spectrum antibiotic and broad spectrum antibiotic. Also discuss the meaning of selective toxicity in terms of antimicrobial chemotherapy. Next objective of this test is microbial resistance to antimicrobial agents. In this objective, state 5 mechanisms by which microorganism may resist antimicrobial chemotherapeutic agents. Also briefly describe R plasmid and name 4 bacteria that commonly possess these plasmids. Next objective of the test is antibiotic susceptibility testing. In this objective, state why antimicrobial susceptibility testing is often essential in choosing the proper chemotherapeutic agent to use in treating an infection. Results. We are going to see how the results will be declared. The point is interpret the results of a Bohr-Carbide antimicrobial susceptibility test when given a Muller-Hinton agar plate, a metric ruler and a standardized zone size interpretation table. So we are going to see first objective of the test in detail, that is antimicrobial chemotherapeutic agents. Very first point is antimicrobial chemotherapy is the use of chemicals to inhibit or kill microorganisms in or on the host. Chemotherapy is based on selective toxicity. In order to be selectively toxic, a chemotherapeutic agent must interact with some microbial function or microbial structure which is either not present or is substantially different from that of the most. Next point is human cells do not contain peptidoglycan and possess eukaryotic ribosomes. Therefore, due to absence of the peptidoglycan and presence of eukaryotic ribosome, or we can say these are ATS ribosomes, the drug shows little if any effect on the host. We can say this is selective toxicity. Next point, based on their origin, there are two general classes of antimicrobial chemotherapeutic agents. First one is antibiotics and second one is antimicrobial chemotherapeutic chemicals. Let's see them one by one. Antibiotics, substances produced as metabolic products of one microorganism which inhibit or kill other microorganism. So antibiotics are the products that are produced by any one microorganism and it will inhibit the growth of other microorganism, and also it can also kill this particular microorganism. Next one is antimicrobial chemotherapeutic chemicals. These are the chemicals synthesized in the laboratory which can be used therapeutically on microorganisms. Next objective of our test was microbial resistance to antimicrobial chemotherapeutic agent. So let's see this in detail. Very first point is a common problem in antimicrobial chemotherapy is the development of a resistant strain of bacteria. Most bacteria become resistant to antimicrobial agents. These changes in the bacterium which enables it to resist the antimicrobial agent occur naturally as a result of mutation or genetic recombination of the DNA in the nucleotide, or we can say as a result of obtaining plasmid from other bacteria. Next point is antibiotic susceptibility testing is usually done in the clinical laboratory to determine which antimicrobial chemotherapeutic agent will most likely be effective on a particular strain of microorganism. Other objective is antibiotic susceptibility testing. The point is, for some microorganisms, susceptibility to chemotherapeutic agent is predictable. It is for some microorganisms, not for all microorganisms. However, for many microorganisms like Pseudomonas, Staphylococcus aureus, and gram-negative enteric bacilli such as Escherichia coli, Serratia, Proteus, etc., there is no reliable way of predicting which antimicrobial agent will be effective in a given case. Several tests may be used to tell a physician which antimicrobial agent, this is not agent, this is agent, T is missing over here, is most likely to combat a specific pathogen. There are many tests available. Few of those tests given over here. The first one is the dilution test. Next one, the agar diffusion test. And the third one is automated test. So first one, tube dilution test. This diagram is showing tube dilution test. You can see the procedure. These are organisms. In these organisms, no visible growth. These organisms grown to standard turbidity in broth. After growth, you can see this looks something like this. Tubes with increasing drug concentration inoculated with a standard number of organisms. You can see these tubes from line, and these are inoculated with a standard number of organisms. So the procedure goes here. You can see the concentration of this tube is 0.5. This has 1, 2, 3, 1, 2, 4, 8, 16. It is doubling continuously. Doubling concentration is taken. Here you can see the growth is very low, or we can say no visible growth. And when these all are plated on this particular plate, then there is no growth when plated, and microbial count is 16 micro per ml. This is tube dilution test. Dilution is done continuously. Dilution is a process in which continuous decrease of organisms occurs from one tube to another tube. Next test is the agar diffusion test or Bauer carbide test. You can see these are organisms grown on the agar plate, and this represents agar diffusion test. Third one test is for the antibiotic susceptibility testing is automated test. Here comes the procedure for the test. First point, take 3 Muller-Hinton agar plates. Label 1 as Streptococcus aureus, 1 E. coli and 1 Pseudomonas aeruginosa. Using your wax marker, divide each plate into thirds to guide your streaking. Next point, dip a sterile swab in the previously standardized tube of Streptococcus aureus. Now squeeze the swab against the inner wall of the tube to remove excess liquid. Third point is streak the swab perpendicular to each of the three lines. You have to streak something like this, and you have to streak your swab perpendicular to these three lines. Perpendicular means this is making an angle of 90°. This streaking is perpendicular to its three lines drawn on the plate overlapping the streak to assure complete coverage of the entire agar surface with inoculum. Fourth point is using the appropriate antibiotic disc dispenser. Place gram-positive antibiotic containing disc on the plate of Streptococcus aureus and gram-negative antibiotic containing disc on the plates of Escherichia coli and Pseudomonas aeruginosa. Next point, make sure that one of each of the antibiotic containing disc in the dispenser is on the plate and touch each disc tightly with sterile forceps to make sure it adheres to the agar surface. Next point, incubate the three plates upside down at 37 degrees centigrade until the next lab period. Next point, using a metric ruler, measure the diameter of the zone of inhibition around each disc on each plate and millimeter by placing the ruler on the bottom of the plate. You can see this diagram is showing the measurement of zone of inhibition. You have to put your metric ruler like this, and around each disc of plate you have to measure zone of inhibition. Next point is determine whether each organism is susceptible, moderately susceptible, intermediate or resistant to each chemotherapeutic agent using the standardized table 8b. This is shown in diagram. And you have to record your result. We will show you a result table. Next point in the procedure is antimicrobial susceptibility testing by the disc diffusion method, and this is modified by Kirby-Bauer and antibiotic profiles. First of all, we will see purpose of procedure. The purpose is to test isolate bacteria for its susceptibility to antimicrobial agents. Specimen requirements. In general, a pure growth of the isolate will work as a specimen. Equipment required will be nichrome wire loops and sterile cotton swab. Reagents required will be Mueller-Hinton agar plate, brain heart infusion broth, and this will be 5 ml amounts. Appropriate antibiotic disc or dispenser. McFarland standard, that is 0.5, and pure culture of the test organism. Method of test. First is preparation of media. First point, prepare sufficient amount of MH and BHIB and sterilize by autoclaving, and see appendix for preparation. Next point, pour of MH medium. You can see the full forms: Mueller-Hinton medium, that is MH, and BHIB, that is brain heart infusion broth. We will use these media in short throughout the procedure. So you can see you have to pour off MH medium into sterile petri dishes at 4 mm depth and let it stand at room temperature to solidify. After preparation of media, you have to prepare inoculum. So let's see preparation of the inoculum. Inoculate the test organism into BHIB tube. Pick 3 to 5 well isolated colonies. Incubate at 37 degrees centigrade for 4 to 6 hours. Adjust the concentration of the inoculum by comparing the turbidity of BHIB tube to that of 0.5 McFarland standard. Next is preparation of the plates. The point is, dip a sterile cotton swab into the adjusted inoculum tube and drain excess fluid by pressing the swab against the wall of the test tube. Next point, hold MS plate, that is Mueller-Hinton plate, half or partially open and streak the plate using the wet cotton swab covering all the area even at the slides. Next point is, place the plates aside for about 10 minutes. You have to allow the inoculum to dry. So the next point is using a sterile forceps or a needle. Apply a set of suitable antibiotic discs. 5 to 6 discs for each plate or 8 discs if you use the automatic dispenser. Next point, let the plate stand for 10 minutes, then incubate in inverted position at 37 degrees centigrade for 18 to 24 hours. Next comes reading and recording the results. Using a ruler or caliper, measure the zone of inhibition around each antimicrobial disc and record it. You have to record the zone of inhibition for recording and reading the results. Next comes interpretation of the results. How you will interpret the recorded results. So for this, consult the special chart provided by the manufacturer of the antimicrobial disc. You have to consult that one chart and interpret results as sensitive, resistant or intermediate. You have to interpret the results as these microorganisms are sensitive to antimicrobes, antimicrobial agents or resistance, or these are showing an intermediate sensitivity. You can see what is the procedure for the interpretation of the results. These are discs, these are your plates, and these are tubes in which you have started the test of dilution. This plate is showing the zone of inhibition. This is ruler and you have to measure the zone of inhibition for the results. You can see these tubes will look something like this. And interpret the results following steps of the procedure and record your results in the tables. Interpretation of the result totally will depend on the procedure, what procedure you have applied for the test, and you have to record your results in the table or tabular format. So thank you very much guys for watching this session. Follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp and also on LinkedIn, that is a major job platform. For latest videos and updates, you can subscribe ITLS Academy on YouTube. You can also get app of the ITLS Academy on Google Play Store so that you can easily access all the informations, all the updates easily. Thank you for watching. If you like the video, then like and share and also subscribe this channel. For any help or query, you can call us on our helpline number 7080833450. You can also reach us on www.itlsacademy.com. Thank you very much again for watching this session.